So for those of you who don't know me, my name is Christopher Wells. I am the Vice President for Public Affairs at the Personalized Medicine Coalition. And I'm fortunate enough to be able to introduce Meg Terrell this morning. For most of the people in this audience, Meg is a person who truly does not need an introduction. She has been with us now for, this will be the 4th year. In the first year, in 2016, she did an interview with Daniel O'Day, who was then the CEO of Roche Pharmaceuticals. And she's been with us ever since, interviewing Stephen Ubel from the Pharma Trade Association, as well as several panels of innovators, payers, and providers. And perhaps her most memorable performance last year, she was 8 months pregnant and joined us anyway. And I recall her telling her physician back home that there's a lot of good doctors in Boston. So she felt pretty good about it. So we were thankful to have her this year with a little bit easier on her travel schedule. Of course, her participation in the conference is a small part of her professional life. Meg is biotechnology and pharmaceuticals reporter at CNBC. Meg has covered a lot of important issues. She's tracked the development of new drugs for Alzheimer's, cancer, and rare diseases. She's investigated the utility of genomic sequencing, something everybody's interested in here, by chronicling her own, the sequencing of her own genome and what that experience was like. And she's been reporting extensively on the drug pricing and access issues that have been a mainstay of the political dialogue in the recent years. So Meg reminds me of a saying I heard when I was getting my degree in journalism in undergrad, which said, ratings do not last, but good journalism does. And I think it's safe to say Meg is a person who has always pursued the latter, and we're just very thankful to have her here. So without further ado, Meg.
Bring Dr. Stoffels right up with me because we can just jump right in. You know, I actually had a weird experience after getting my genome sequenced in childbirth, to kind of bring two of those stories together. So I had my genome sequenced as a healthy person, just for, you know, a story to see kind of what it was like and you know, whether I could use the results. And it turned out that I had a one thing, factor V Leiden, just one copy of the mutation, which means that my blood clots a little bit faster, I guess, than other people's. Okay. And so I shared this information with my doctors. I didn't mean to tell you guys this story. It just reminded me during the introduction, and it kind of leads into what I want to talk with Dr. Stoffels about. And after I had the baby, they put me on a blood thinner. And I just kept saying, why am I on this blood thinner? I don't have a clotting disorder. And so they made me take it for 2 days, and I kept questioning it, and the doctors kept being like, well, are you refusing your medicine? You know, it was a very sort of weird thing, and I'm like, well, I just want to make sure you guys like understand the genetics and that this is really what I'm supposed to be doing. And then finally they decided I didn't need to take it. And I talked to my geneticist afterwards, Dr. Green at Harvard, Robert Green, who seems to sequence all of the journalists who get their genomes sequenced. And he was like, this is a problem, that you got this blood thinner, and it doesn't necessarily indicate that we are using this genetic information correctly. When you have your genome sequenced as a healthy person, doctors don't always know exactly what to do with that information. And he said this could lead to problems with people bleeding, you know, because they got unnecessary treatment. And so that leads me into the first question I want to ask Dr. Stoffels, who I don't think needs any introduction. He's the Chief Scientific Officer of Johnson & Johnson, which had a research budget last year of $10.8 billion. Is that what you spent on research?
Yes, we spent 10.8 on research, yes.
So that's more than a quarter of what the NIH spends every year, about $39 billion. So you have quite a lot of power in terms of your research-making decisions. You've been at J&J for a long time, but you started your career as a physician in Africa, focused on HIV and tropical diseases research, so an incredible wealth of experience and knowledge that you bring to this role. So I wanted to talk about what you see as idealized medical treatment. In a perfect world, you know, what does my experience look like when I go in to have a baby? What does everybody's experience look like with the healthcare system?
Well, idealized is healthy longevity is how can we prevent and treat as many possible ailments of diseases as early as possible, either prevent, intercept, or make sure that when we find them, we treat them and we cure. I think if you look at the past 20 or past 100 years, then we have been able to get rid of many diseases in the world, yeah, through vaccines, TB, through antibiotics. It's not under control everywhere in the world, but if we were here in 1930, TB would be the topic of the meeting because big problems, many people, many families would have. But those diseases are now all gone because we have good drugs, high blood pressure, most of us elderly people and myself, we are an anti-cholesterol medicine, so you prevent, over time we have been able to prevent a lot of diseases and make sure that they don't cause significant challenges. That is the ideal way for me to how treat. Simple healthcare system, access to everybody, so that everybody can benefit from the healthcare system, but at the same time not wait until we have late stage cancers and then can't treat and can't cure people anymore. So more early stage determination of risk, more early diagnosis, better targeted therapies so that the right people get the right therapies, prevention if possible with vaccines, if not with treatments, and make sure people have a healthy, long life. Mm?
Well, so that's easy.
It's not easy, but it's a goal and it's a vision and a mission, yeah?
Where would you say we are, in terms of reaching that? If that's 10, where are we?
Well, it depends where you are in the world, but let's say right in the middle. We have gone a long way already. We have a lot of platforms which we can use now. If you go 100 years back, diagnostic drugs were not there. Today we have a lot of diagnostics. We can treat a lot, we can prevent a lot. But the most complicated and challenging diseases are still need to be tackled. If you look at dementia, if you look at depression, if you look at many of the cancers, if you look at a lot of orphan diseases, while a lot of platforms are there to early diagnose, treat or cure, a lot of work is still needed to get done. And we are in an incredible time at the moment with new discoveries in the last several years. If you look at immunotherapy for cancer, changed the way cancer can be treated. If you now look at CAR T cells, the cell therapy, the first indications that you can use cells directed to cancer with spectacular results. So it are all these first gene therapies where now we can introduce or we can treat genetic, so these platforms are there. But like always with platforms, and that's the benefit of being 30 years in pharmaceuticals, we started with small molecules. We became very good at it, making very good medicines. It took us 20, 30, 40 years to get there. Then we got to antibodies and protein medicines. Again, 10, 20, 30 years, and now they're widely available in many different diseases. Cell therapy, we are at the beginning. Gene therapy, we are at the beginning. And once the platforms are established, the experience is there, the safety and efficacy, you know how to use the tools to direct them to biology. At that moment, they become fully functional in large-scale healthcare implementation. And that's still, we are only at the beginning of the most breakthrough platforms at the moment.
Do you see those timelines shortening with new technologies?
Yes, I see them shortening because of a lot of different things. We have now access to data, and I think the access to data where we learn about what works, what doesn't work, to electronic medical records, faster clinical trials, virtual clinical trials. We have so much access to data now that learning about how platforms behave in people or drugs behave in people, we know much faster. Artificial intelligence to have much more insights, combine diagnostics, molecular diagnostics, clinical outcomes, and all the different permutations on a large scale gives us much more information. So we can decide faster. We can also, not to forget, the regulatory authorities have changed quite significantly with the number of breakthrough designations now. If you get really material innovations, I think at J&J we had more than 10 breakthrough designations. You see the speed which you can develop new medicines is accelerating. So more and more new drugs will become faster, new platforms will become faster to accessible because of all all these information, insights, regulatory pathways, accelerated clinical trials, and fast experimentation.
I want to ask you about data. Companies like Regeneron and Amgen, they've partnered or acquired companies that are doing vast amounts of genomic sequencing, pairing that information then with electronic medical records and other information. Is J&J tapping into genetic data in a similar way? Where do you get your data?
Yeah, big time. And I go back to my own experience where I was a significant part of the HIV medicine discovery and development, but I started with a data company. I had with Verco, we sequenced 600,000 patients, yeah? And that information, we phenotyped 120,000, we combined it with clinical data and learned from that how to get to the best new medicines for patients faster. And that learning if we now do every therapeutic area of every disease area in our organization uses the collaboration on data with different organizations to determine biomarkers, early detection, response parameters. All that is part of our is a second nature. It's part of who we are and what we do. We have a lot of collaborations, but we never bought a big company or never acquired a big company. But for safety today, we have access to de-identified data of more than 1 billion people in the world.
A billion?
A billion, yes.
How do you have access to that? Through all of these different partnerships?
You do different partnerships, academic, there are coalitions on that. So today, to learn about safety of your medicines in the world, you go through, of course, the direct reporting to you from patients, physicians, from hospitals, but more important now is access through data systems, and they're all de-identified. So we don't know who they are, but we can see if one of our medicines has a side effect, we can, through our data partnerships, can find that very quickly. Mm-hmm. And that also helps to identify activity of medicines. We do a lot where we do data access to learn outcomes in the real world from medicines. So data have become a critical platform in every company who wants to be successful. Mm-hmm.
And you mentioned AI. How intelligent is it right now? How helpful is it, and how are you using it?
Well, AI needs a lot of data to be trained in order to be able to make to do the decision making. So it's let's say the early days of being really relevant, but especially at this moment in discovery sciences where a massive amount of data are being generated, it's absolutely helpful to go through the better molecules, the better targets, it starts to be integrated as part of a data management or a data access almost everywhere. To real practical clinical implementations, the most advanced there are in imaging world, in radiology, CT scans, in anatomopathology. There AI is now routinely practical. In medicine, it starts to become part of the world. Yes.
Mm-hmm. Let's talk about the successes in personalized medicine and how to translate that into other therapeutic areas. Obviously, cancer has been an area that's just out in front of everything else, it seems like, in tailoring treatments, whether it's using a patient's own cells or just identifying what's driving their disease. Where do you see that progress in other areas like neuroscience, other areas where J&J is operating, and how do you catch it up?
Yeah, we try to learn about in every field, whether it's in psoriasis or in inflammatory bowel disease or in cancer or in infectious diseases. We try to learn everywhere response, non-response. Can we find the parameters which determine that to better target therapies to the right patients? And so recently we brought a new medicine, Balversa, for bladder cancer, which only works if you have a specific EGFR, it's an EGFR, targeted EGFR medicine, but if you have a specific mutation, yeah, and more we go to combining specific diagnostics with drugs. In mental health, what are the parameters, and there are different types of markers predicting whether people will respond yes or no in depression. And that is more from the data side. Other is more biological. The first field, let me go back to my background on HIV. We, in very early stage, did resistance testing, viral load, and combination of therapy. Almost every patient had their individualized therapy. And because the patient demanded it and fought for it, it was very fast. And so it developed very fast. If you now look in oncology, it's going in the same direction. The molecular diagnostics will be able to determine which drugs are good for you as a patient. Response rates will be able to be measured very quickly. And adaptation of therapy will go, and every patient will become a single patient type of trial. It's not a trial, it's implementation. So while initially it's difficult to get done, and it are lab developed tests, and before they are PMAs and can get onto a large scale into all of the laboratories, it takes time because these tests needs to be developed. But as they show their clinical benefit, they will become part of mainstream. Who would have thought that today genetic testing for XDR and MDR-TB is now available all over Africa, all over Asia, in most of the laboratories to determine which medicines need to be given for TB. And that is a result of all this technology development, resulting in a simple assay which can be done in the field with molecular diagnostics to be able to determine what therapy people have to have.
Tell us about how you decide what to focus on in terms of developing new medicines. I assume obviously you follow the science, but also there are some areas you've been working on for so long, HIV vaccine being one of them. It takes a really long time to develop things like that. And it's not always the thing that you'd say makes the most sense if you're driven completely by the capital markets, which sometimes it seems like pharma pipelines are. That doesn't necessarily jive.
No, you're probably going to be surprised from my answer, but it's mostly driven by the passion of the scientists and the people in our organization. So, and the availability of new scientific platforms. We are, as a large company, I see ourselves as the connector between good new science and applicable science and specific diseases we can solve. But you need to have very dedicated teams who have insight and know-how for many, many years. You don't do a market study and say today, hey, we are going to, because the capital markets like it, we are going to shift our focus. That's not how this works. It are teams who have 5, 10, 15 years of experience and the passion in a certain area, who know the entire field, who can decide that science, if we use it, will come out in 5 to 10 years with the best medicine. Pharmaceutical development is more about insight in the future on what you can do with this science than learning today on what you can do with the science, because the world is evolving. If you look at multiple myeloma, and we work in that field for 15 years, now we have CAR-Ts, which probably will change that field. How are we going to make the next generation medicine 5 years from now, which is better than what we see now coming into the market? And that only works with very passionate scientists, physicians. That's one, the availability of good science. And of course, you need a problem to be solved. So you have the law of the maximal achievable benefit in a certain disease with a certain platform. And the pharmaceutical world follows first generation, second generation, third generation. Typically you can get to better medicines, but then you achieve the maximal what you can do in that disease. Look at very simple example, hepatitis C. If you can cure with one pill in 3 months hepatitis C, that's solved. What are we going to do? You go to somewhere else. Okay, now hepatitis B, or you put your people on other areas. So you have to, at a certain point, switch. But it's always driven by the passion and the know-how of our people, availability of good science, and a good, important problem to be solved. Years of life, quality of life. You have to be able to measure the benefit, first in years of life, of quality of life, before you make a business plan. That is the most important metric. Can you change life? years and quality. And if you can do that, you can start calculating, is it worth investing in it?
So then once you've made that decision and you have to figure out the business plan, how would you assess the business plans right now for personalized treatments, CAR T and gene therapy in particular?
Yeah, I think financial, the way we do the business is organized today, or the healthcare system is organized today, should not be an inhibitor to get to new breakthroughs. If today with CAR T you do one administration and the patient will benefit for 3 years or 4 years or 5 years, we have to solve it different than saying that's not fitting our business model. We have to find a business model where we can bring that in. And we all lease our cars, or have our mortgages on a house. So you could pay for diseases also over many years, even if it is one time administered. The only thing we have to do there is start working on a business model which can accommodate that type of therapies. So you have to, today we do CAR Ts, we are working in gene therapy, and the financial models behind it, don't worry us, we'll solve that, or we'll solve that with the payers in the healthcare system. We have to, because otherwise it stifles innovation and we won't go anywhere. And that's, don't put the financial models and the business models of today first before you invest. Make breakthroughs and adapt the business model so that they can accommodate the new technologies.
So what you're seeing so far in terms of the CAR Ts that are on the market, the few gene therapies that have been introduced, is it working? Or do you think that you're going to have to change it as more are introduced?
It will be need to be adapted. The one-time very high payments are very challenging for a healthcare system, or very challenging for society. We have to find better solutions for that. Yeah, and as it will become much more. At the moment, it's one-off here, one-off there in an orphan diseases, but it will become much more regular now that maybe 5, 6, 7, up to 10 of these therapies come into the market every year. So we'll have to change the way we think about that. And industry has the role to work with the payers, work with society to find these different models. Yeah.
I want to ask you about the tension between the way the public sees the industry and what you're describing as you're driven by the passion and the ingenuity of the people who work for you. So people go to work in this industry every day thinking they're doing great work. They're doing some of the most important work there is. It's harder than rocket science. But the public, in a recent Gallup poll, said they hated pharma more than they hate the federal government, the least popular industry in the country. Yeah. J&J is dealing with some specific problems, the talc powder and the disagreements with the FDA over finding asbestos in one bottle, the opioid crisis where J&J is offering a settlement of $4 billion. How do you just go to work and kind of grapple with that tension of how the public sees you and the importance of the work that you're doing for society?
Well, I'm there, and I can tell you that the motivation within our organization at the moment for solving the next big problem is incredible. Yeah? The motivation for people to be part, as human beings, to be part on changing the future of healthcare, whether it is with an HIV vaccine, or we are now introducing a new Ebola vaccine, or new TB drug, or new cancer, or a new CAR T. It's such a magnificent space that it doesn't impact the way we think about the future. We have to deal with the way we are perceived outside, but the way we partner with biotechs, with academia, inside our organization, with diagnostic companies, we are the driving force of changing the future of healthcare by the way we bring you, by the science. The early science, we are the translators into real application for humans. I agree. We'll have to do better on how we are perceived, but it's not going to change the way we think about science, and the way my teams in the organization, as scientists and physicians, will continue to work. And you've seen that in several of the meetings you had with some of them. So it's an it's a problem on the pricing of medicines that the public perceives as very high, and they actually probably are. The investments we do are, like you say, 10.8 billion in investment in R&D every year. That is quite substantial. There are very few industries who can do that, and it's because we are innovative. We bring innovative drugs, which we bring into the market value-based, because almost everywhere in the world, the value proposition we make, we negotiate and talk with the payers. It can be perceived as high, but if we calculate outcomes-based measures, then almost all the time now, it is in a determination in the discussion with the authorities. And that we'll have to do better, but I think we will get there.
Have you, you've been in this industry for a long time, have you felt like it's changed the public's perception of the space? It's always been a tense relationship, but is it worse now?
Yeah, it always has been tense relationship. Having been in the industry for 30 years, in the first days of HIV, we had the same debate. The first days of hepatitis C, you have again the debate. Bringing new innovation at a high cost is where many people need it, is always been the red flag on hitting us. Yeah, but in the meantime, if you look at what we have contributed to HIV, to life expectancy, today, when I started in Africa, life expectancy was 6 months there, 2 years here. Today, life expectancy is normal, or maximum minus 2 years, 30, 40, 50 years of life expectancy, because the research of all of our companies have done and the combinations we bring to patients. So. And it's now with tiered pricing available for $70 to $100 HIV. People are treated in the middle of Congo, middle of Africa. Everyone is treated. 30 million people now get therapy. So you can't deny the contribution of what industries like us do. And of course, the way we have to create a business model. Typically 10 years of even shorter after approval, we lose our patents. We have to recover our cost and the benefits within the 5 to 10 years after the launch of a product. And that's how our business model works. Yeah?
Mm-hmm. Well, sometimes, I mean, oftentimes companies find ways of extending their patent protection for many, many years past that point.
Yeah. But very often time we also lose it, yeah, much earlier, yeah. Or we fail a drug which we have invested a few billion in, and it hits a safety, and we have to take it from the market. So we have the positives and the negatives in that space, yeah. And many drugs only are short-lived because the next generation medicine is coming much faster than you expected, and you benefit 2-3 years, and then it's gone. It's not the first time it happened to us too, that we have a medicine which only exists for 3 years in the market because the next generation is there. More and more, by the way, is happening because innovation is going so fast.
It's going faster. I mean, that's good news for folks, but it makes your business models more difficult.
More difficult, yes.
Hepatitis C was a fascinating, I think, market study of that phenomenon, probably.
It was a fascinating, fast. Once we could culture hepatitis C and could screen, everyone and their brother-in-law could make an hepatitis C drug. Yeah, so it was very, very fast. And at a certain point, you had like probably 100 new hepatitis C medicines somewhere in the market. And in the end, 2 or 3 made it because it was much more simple in the end than everyone expected. And that gave a very fast turnaround of the market, and ultimately a few remaining players with relatively low price for a cure now. So the market works there. Yeah?
Well, so when you mentioned everybody and their brother or sister being able to make something, that makes me think of immuno-oncology. How would you assess that marketplace and how many different shots on goal there are following similar targets?
Yeah. Well, immuno-oncology has been a breakthrough, yeah, for cancer, and many companies will develop one because you have to have it as part of your combinations. Yeah? And so it's all about how can you get to combination therapy. And so many companies have one in development to be able to use their own in combinations, because the combinations are going, if you have to pay for all of the different combinations is going to be very expensive, first in development, but also ultimately in the market. And that's why you will see a lot of, and I think it's good that a lot of immunotherapies are developed, because like in hepatitis C and in other fields, it will bring prices down over time and much broader available, and many more combinations will emerge. So I think it's a good thing. And there is also a market mechanism that once it is not, doesn't give any return anymore, it will stop. Yeah, so you have to give up. Yeah, so the market mechanisms work there.
As you look out into the world and look at potential partnerships in academia or in small biotechs, how much exciting science that is different from the pack are you finding there, especially in the immuno-oncology space?
I can't particularly answer in the immuno-oncology space. Very broadly, if you look at what we learned in CAR-T and different type of is massive. Yeah, and that if you learn now or see now what is happening in China, the number of CAR-T companies which have emerged. Also here, many academic centers were able to do that. So it's going to evolve very fast. The future of that, in the end, it will have to get to a number of platforms which emerge from that, because you can't have 100 platforms in CAR-T hanging out in a hospital. You have to, will have to either with industry or in collaboration, will have to come to how is that becoming more routine. But CAR-T, in my opinion, is also a very important proof of concept. Which will lead to new type of cells, which leads to new type of future cell lines, which will be much easier and probably off the shelf to have the same effect as you have today with CAR-T. So it's a proof of concept. It will work. Many patients will benefit from, but very quickly will evolve into next generation. And that is the good thing about all these discoveries, that they will transform cancer. They have such a good benefit that the attraction to invest in it is very high, and the next platforms will emerge very quickly. And all of us will benefit from that.
I think we'll get to talk about this a little bit more on our next panel. So I'll just ask you one last question before we then shift. And Dr. Stoffels, we'll be sticking around and we'll be taking questions in the next one, I think. So you'll get to ask Dr. Stoffels questions. But you mentioned you're driven by the science and the passion of the people who work for you. What are you most passionate about? I know it's hard to choose. Tell us about one really exciting project you have going on right now.
What I'm most passionate about, well, I'm an infectious disease person, so if I would have my dream realized is have a vaccine for HIV. Because of the still tremendous impact on health in the world. If you go outside of the US and Europe, but even within in our countries, the long-term pathology of HIV, especially in the developing world, is still an exceptional negative contribution to the future of these countries. Having people taking 50 years their pills, it's a challenge, and we have to get to different solutions. And that is, a 30-year-long passion from my time in Africa, treating patients in hospitals, to where we are now, being able to treat them very well, but the challenge of treating 30, 50, 40, 50 million people for the next 40 years is a huge challenge. And so we have to stop the transmission of the virus, and so that over the next 50 to 100 years, the disease can get out of the world. And that's only possible with the vaccine. So
how far away do you think you are?
Well, we are 2 years away from the outcome of our clinical trial, but that doesn't mean that we will get to a result. But we learn definitely. Every experiment you do, you always learn, and you learn from the successes, you learn from the failures, and that's almost in every space that you have to learn from the failures. Don't give up. Continue to go to the next step and incorporate the learnings and go forward. And that's what we do.
Dr. Stoffels, thank you so much.
My pleasure. Thank you.
I think we stay up here now.
Yeah.
So we're fortunate now that Meg is going to carry on and continue to moderate the next panel. So we'll have the panelists come up and I'll let Meg get started with that.
So we'll continue this conversation with a great panel of folks. Prospecting the pipeline, exploring the implications of a biopharmaceutical pipeline dominated by personalized treatments is our topic. So we're joined by Dr. Alex, do you pronounce it Vadas or Vadas?
Vadas.
Vadas, who is managing director and partner at LEK Consulting, who I was reading, you've done a lot of work in companion diagnostics. So a lot to talk about there. Dr. Donald Siegel's director of the Division of Transfusion Medicine and Therapeutic Biology and director of the Clinical Cell and Vaccine Production Facility at Penn's Perelman School of Medicine. Dr. Harpreet Singh is the CEO of Maddox, which is a clinical stage biopharma company focused on T-cell redirecting immunotherapies for the treatment of cancer. So a lot of the things we were just talking about, we'll get to dive into more. And of course, you guys remember Dr. Stoffels. So we'll just get started. I mean, the question that we started with Dr. Stoffels, I'd kind of like to ask you three as well. Where is the idealized way of us getting treated in the areas where you guys are working, and how close are we? Why don't we start with Dr. Siegel, who's in the academic and research stage of that?
Sure. Well, first, I'd just like to thank the organizers for inviting me today. I got my PhD at Harvard between 77 and 83 and haven't been on this street for about 40 years. There's a few memories. I used to play in a music group which rehearsed across the street in Boston Latin. Then I had my car, my Volkswagen Rabbit, had a boot put on the tire by the police right outside here on the street. And then down the street in Building A of the medical school was where I was rejected from Harvard Medical School. So there's just a lot of memories that are just floating in my head here. So it's a delight to be back here. Yeah. So actually, after I was rejected here, I went to medical school after getting my PhD at Penn and never left Penn. I've been there for 36 years and have seen, so I went to medical school there, got interested in clinical pathology, which is not the microscope kind of pathology, it's more of the clinical laboratory pathology. Got interested in transfusion medicine, which is the therapeutic part of pathology. So I run the division of transfusion medicine that incorporates the blood bank, the apheresis unit, the stem cell lab, which is where cells are processed for doing bone marrow transplants. And then what developed over the years was creating a laboratory for engineering cells, which led to CAR T therapy. And so the philosophy we've always had is that this CAR T business is just the 21st century version of transfusion medicine. There was some discussion yesterday morning with Carl June, whom I work with, about regulatory issues and so forth. And I think that kind of stuff has all been worked out in the blood bank field. We transfuse 60,000 bags a year in our blood bank alone, and we get it right. And so I think a lot of the reasons I think why we were successful at Penn is because we didn't look at this as a completely new thing with absolutely no way of going about structuring it, but that it's an extension of how we've dealt with the initial personalized medicines, which was transfusion, and before even personalized in terms of blood type, in the 1500s, people would transfuse an animal's blood into someone based on what you were trying to treat. So if someone was very aggressive, you'd use blood from a calf. If someone was kind of crazy, if someone was very docile, you'd use blood from a bull. I think that was the first example of personalized medicine. This is in the 15th and 16th century.
So were they using companion diagnostics for that?
Well, dogs are companion animals, I guess, so that's the companion part of it. Yeah. So I guess to answer your question, I sort of have three roles that I serve, three hats I wear. One, I have a research lab where I develop the parts of new CAR T constructs to develop new CAR Ts. But from a clinical side, I'm running the GMP facility that produces our own CAR Ts that are being used in over 20 different trials. But then I think maybe more germane to this is the fact that now that 2 of the CAR Ts have been FDA approved, we, like many other academic centers, have physicians who prescribe these products. And so across the street in the hospital, we are participating in this how you get these manufactured drugs to patients. So in other words, we had to have our own apheresis unit, which is where patients come to have the raw material collected. We had to be approved by Novartis to collect it, even though we had invented the process. But it had nothing to do with it. We had to go through the whole thing. And then we had to have our lab approved for freezing the cells, and then send them to New Jersey where the product is manufactured and then sent back to us where we then store it until we administer it to the patient. So I've seen, and that whole part is what's kind of going crazy right now because every company is coming in with their own process for how you do that. And so our stem cell lab, which was used to processing 300 or 400 bags a year for stem cell transplants, now has to learn the process that all these different companies have agreed to with the FDA for how cells would be collected, whether they'd be frozen or sent fresh to the company. We have a whole section of our laboratory now is just filled with shippers, liquid nitrogen shippers to receive things coming in, send things out. And this isn't just us, this is the way it's a whole new kind of thing happening all across the country.
Oh, it looked like you were wanting to...
Well, it is absolutely a challenge to industrialize such a process, but once you get approved with the FDA, every patient is a batch. You have batch records, and you as a company are responsibility for the outcome of that, and that's where it is a very controlled process from the A to Z, because nothing can go wrong, because as a company you're accountable for the product which is infused, and unfortunately that brings a lot of quality challenges, of quality control challenges in the whole supply chain.
Dr. Singh, why don't you tell us a little bit about what Maddox is working on, and how you see yourself getting to, as we were talking about with Dr. Stoffels, anticipating what is coming to the market in the near term and making sure that you're then ahead of that. I'm sure as a CEO of a biotech company, that's what you think about constantly.
Yeah, constantly. That's true. My history is not as colorful as Dr. Siegel's. I was not rejected at Harvard, but never applied. I'm an immunologist.
You're smart.
I'm an immunologist trained at the University of Tübingen in Germany. And I worked with Hans-Georg Rammensee, and Rammensee in the mid-90s did a fantastic discovery, and that was to show actually how the immune system displays itself, actually displays what's friend versus foe. Sort of how tumors, how viral infected cells display to the immune system, whether they are intact or not intact. And that's done through peptides that are presented on so-called edge layer receptors. This was such a fundamental breakthrough, and every company nowadays that is working on T-cell therapies is basically developing therapies that discover that basic mechanism. I was very fortunate being in that lab, and we started looking at these peptides on tumors and simply checking what's on cancer tissue presented in terms of peptides and not presented on normal tissue. And started doing this very systematically, which led to the foundation of Maddox over 15 years ago, and starting that exercise at that point, and now having done this 400 million times, and really mapped out what we call the cancer immunopeptidome as really as a starting base for that. A number of interesting experience. Number one, we started believing in T cells 20 years ago. I remember actually going to pharma companies, including J&J, it wasn't Paul, and telling them T cells can make this work, and the answer was, Harpreet, come on, T cells are homeopathy. They cannot work, they're just too mild. And it's just wonderful how the field has evolved. It wasn't your fault, obviously. but it's wonderful how now we actually see the immune system being one of the most powerful weapon in their arsenal against cancer. The other part is actually when looking at tumors 400 million times to appreciate how different they are, particularly solid cancers. Every tumor is completely different. What's displayed actually to the immune system is completely different. And that was really the basis for us to say, let's create something where we can really address that heterogeneity of the tumors. So I created the company together with some fellow PhD students. I was 25 at that point. And we wrote a business plan, and the business plan's title was, in 2004, Personalized Cancer Immunotherapy. Absolutely zero chance to get any venture capital. for something that's not a drug, personalized cancer immunotherapy. I mean, that's totally crazy. We ended up focusing on a few of these targets and developing a drug on these few targets, but we've never really let go. So Maddox now is a company that is globally leading in the space of IO target discovery. And at the base of IO discovery, we are generating a number of products in a pipeline that is dedicated to cell therapies and biologics, some of them to single targets. But the end game that we really see is a patient coming in and having a warehouse or library of 10, 20, 100, 1,000 different drugs that we can apply to that patient in a very personalized fashion. And pharma companies have bought into that. So we raised money through venture capital to fund this, but we've also received substantial funding from pharma companies such like Amgen, Celgene, Roche, Genmab, and others, and have an alliance with MD Anderson now how to achieve that.
So you have a few different approaches. I was looking in your pipeline, and one of them I think is really interesting is this idea of an off-the-shelf donor cell version where essentially you'd have this, as you were saying, you'd have all these different options on the shelf waiting for people, but they'd actually be donor cells that were targeted against different cancer targets that you'd have waiting and ready to go. How do you see that sort of balancing with the other approaches to T-cell therapies?
So we have 3 different flavors of T-cell therapies, and the reason is very simple. I mean, I could pretend that we have an exact plan, but the reason is we don't know which of these will truly work out at the end. So we're basically having multiple shots at goal. We have autologous cell therapies that we know principally work, but we still have to work out the business model. Is this really, are the costs of goods at the end of the day practical to sort of work out? We have the allogenic platform that you just mentioned, which are so-called gamma delta T cells that we can grow from healthy donors, and actually from one healthy donors could do up to 9,000, can create up to 9,000 doses and put them on the shelf, which dramatically reduces cost of goods. And I think this takes us much further to something that is reimbursable by patients and by society. And then the next step is also going to biologics. So these are basically antibody-like molecules that build bridges between T cells and tumor cells in a way and can be applied very simply. So we believe we have to really tap into the entire arsenal of different drug development pathways to make this happen.
Dr. Vadas, how do you look at the market, where it stands right now, and where you think optimally it would be? What are the hurdles to getting there? Where actually do we stand? Kind of give us a status report.
And specifically, it's associated with the cell therapies or generally for personalized medicine?
How about generally?
Yeah, I mean, well, I'll probably lean on collaboration that we were lucky to do with the Personalized Medicine Coalition, which is a recent publication we did together, which was literally digging into the entire pipeline. And I think there was a mega trend in there that is, you really can't get away from that. 15% of drugs looked at biomarkers in oncology in 2000, and now it's 55%. And I think that is just which basically means that nearly every innovative therapy that's going to launch in oncology will likely have some flavor of a diagnostic biomarker that's tied to it.
I'm actually surprised we're only at 55% right now.
Well, there are a lot of trials in there that are just novel sort of chemotherapy regimens that may be less tailored to these types of approaches, so that maybe repurposing other drugs that are already there, but most of the innovative stuff is pretty much all biomarker driven.
Can everybody hear Dr. Vadas?
Sorry.
Oh, sorry. Can you?
I'll speak louder then. Thank you. Yeah. So.
Or just closer to your microphone.
It's really. I mean, it's going to change the game for everybody. We are now, as a pharmaceutical company, you can't just think about my drug and my biomarker. You have to worry about everybody behind you and everybody in front of you who may impact what biomarkers and diagnostic tests and workups the physicians are going to require for their patients. In care delivery, it's not one test, I'll do HER2 tests and that will tell me what to do. It's going to be a journey of testing that will constantly be required to be deployed. I think the healthcare systems are going to have to adapt in that this is going to become the norm. It's sort of not, well, we know we need to do sequencing in lung cancer because it makes sense there. I think we're going to find that it's going to make sense everywhere. Mm-hmm. So, I think just as we look at what the pipeline will deliver to the market, it is, and this is not just around the specific personalized drugs, but I think just in general around all these precision oncology drugs, I think we are all talking about it, that we need to change, but I think we are just at the tip of the iceberg in terms of the interventions that we're going to have to deliver patients. And this is going to have to change very significantly how we deliver healthcare and the information that we're going to be generating around these patients to help manage decisions.
I want to encourage folks, if you want to hop up and ask a question, please do at any point. Let's just have it going on. We don't have to do them all at the end. So just go to one of the microphones and we'll start integrating into the conversation. This is kind of random, but one aspect that I've kind of been hearing more about, and I'd be curious to hear everybody's thoughts on it, in terms of personalizing treatment and also just generating more data, is the microbiome. How much are you seeing that being integrated into research, into new therapies? And as you're getting data back, and Dr. Stoffels, maybe I'll start with you. Are you looking at it in every different therapeutic area? Are there just certain ones where you are examining it, or do you think it's overhyped? And I think George Yankopoulos told me he thinks it's a complete bubble and it doesn't make any sense. He's of course the chief scientific officer at Regeneron. So what do you think?
Well, I don't think that the impact or the role of the microbiome is overhyped. The challenge of how to make therapeutics to impact it, that's a challenge. And it's very important in inflammatory bowel disease, it's very important in diabetes, in many different roles. It plays a very important role in many diseases, but it's a very complex ecosystem to change something to have an impact. And that's where I think it has shown to be much more complex than we think to make drugs or to make tools to impact it. We have a whole microbiome team who is looking into many different areas, but we don't have very advanced specific therapeutics which we can point to and say this is going to be a therapeutic in the future.
Are you using it to guide, or just are you collecting the information on patients and all of your different clinical trials to use that as just one piece of your understanding of how people might react to different drugs?
It's not in all clinical trials. It's in specific targeted research projects that we collect microbiome information. While we collect molecular diagnostic information in all the cancer studies and most of the infectious disease studies on bugs, we will not on every patient collect information on microbiome. It's too complex and too much work.
Anybody else?
Well, we routinely in our clinical trials collect this. All these trials are at the initially hypothesis-generating trials. So you don't start these trials with necessarily predefined hypothesis, but you collect all the data that you can, and that includes microbiome data. I'm not sure, I think I entirely agree with Dr. Stoffels, how this can be impacted. At the moment, we just want to learn, and so we see this rather from a diagnostic aspect. How does this influence actually the response that we see in our cancer patients treated with various cell therapies? Personally, I believe that maybe just eating yogurt and having an anti-inflammatory diet may be the best way to do that, but that's a very personal statement.
Sponsored by Activia.
No, definitely not.
Just kidding. Anybody else want to weigh in?
So I can't say that the microbiome has played a role in the things we're doing with CAR T cells or other engineered T cells or dendritic cell vaccines, but from the blood banking point of view, we have been engaged with the infectious disease group who want to do fecal transplants. And they obviously need to adopt some of the same safety principles that have been used with transfusion in terms of donor screening, acquisition of samples, processing, storage, giving it to the right person. So indirectly, sort of involved in that, but it hasn't had something to do really with our cell therapy program at this point.
Any thoughts?
Just the simple point, I mean, we have noticed that in some immuno-oncology trials, folks are looking at microbiome as well as other immune markers and trying to make the connection between those two. So I think, and one of the trends we're seeing is a sort of growth in just the number of biomarkers people are exploring in trials. And so layering in immune-related biomarkers as well as genetic biomarkers that are hallmarks of the cancer, and then layering in a microbiome view may actually also, are some strategies we're seeing explored, but I think they haven't yielded to the points of prior. haven't yielded anything clear yet.
I didn't expect to be talking about fecal transplants this morning, but I am actually kind of obsessed with that area, and the.
It's interesting.
I just think it's really interesting, and the analogy to the blood bank, I mean, is such a great one, and so I'm actually curious about your thoughts on, do you see that as is that a model for the potential future of fecal transplantation, and I mean, just what do you see as kind of the future of that space?
So I haven't really thought that much about it as you obviously have, but I mean, I think from what I see going on now, a lot of it is more like in transfusion medicine, we have direct donors, like you donate blood for a specific relative. So I see that more happening now because I mean, there's something about people living in the same house, or I don't know. So there are a lot of analogies, but from what I understand, there isn't some big poop bank being created in my hospital.
There is a stool bank here in Boston. I think it's called
A stool maker?
Yeah, stool bank.
Yeah.
It's really hard to qualify to be a donor, actually. Like,
Well, one of the
it's
problems
really
is
hard.
that it's one of the problems I understand is, about blood donation, you sit in a chair and you stick a needle in. So in this case, you can't, it's not like on demand with the donors.
Unless you're
as
taking
much-
your Activia, I guess.
Maybe, yeah.
Right.
I'd like to follow up on one of Meg's earlier questions and note that there's not a lot of consensus in this country between the parties except on pharmaceutical pricing. And we have now, both in the White House and in the House of Representatives, proposals to import reference pricing to the United States and, in fact, impose some sort of price controls. How that would work out remains to be seen, or if it passes remains to be seen. But I'd like to get a sense from the panel how worried they are about that, or how they think it's a good idea, and what the impact of it would be on innovation, and in particular CAR T therapy.
Okay.
No,
I think when you're just thinking about the cancer application of CAR T and the cost, $400,000 or whatever, others can probably know more about it than I do, but that's cheaper than a bone marrow transplant, which is not necessarily curative. So on the one hand, I don't know that the pricing is that crazy at all. But there are a lot of things that are going to happen that will wind up reducing the cost. One of them is sort of was alluded to here, allogeneic, where you don't have to make the drug one by one for each patient. It'll become a little more impersonal medicine where you have some healthy donors from which you can make a lot of CAR T that can be used off the shelf. That, of course, requires some technology to make the cells compatible with anyone, which involves having them not be rejected by the recipient and also not having them attack the patient. And so we have a trial that's about to start where we use CRISPR to knock out things in donor cells so as to make them compatible. So once you can then scale up, that would reduce the price. It also it makes the therapy available to the patient exactly when they need it instead of having to collect cells from them and manufacture it. The other issue that it alleviates is the fact that the raw material being collected from patients are not healthy people, and the quality of the final product is very dependent on the quality of the cells you collect from the patient, and these patients may have been through lots of chemo and so forth, and so allogeneic gets around that as well. But we're also making strides in reducing the time of growing the cells in the lab from maybe about 10 days. We hope to get it down to 5 days, and maybe even shorter than that. And with the idea that have the cells grow in the patient rather than grow in a bag in the lab where they may kind of poop out. And of course, if you shorten the manufacturing time, then that's going to decrease the cost as well.
Dr. Stoffel, so how do you look at the reference pricing proposals?
Well, we stand by the principle that and if you look at the last 20 years, as an industry, have worked very hard to go from incremental next generation type of medicines to much more transformational, very impactful. And we stand by the principle of value-based pricing in discussion with payers and the government. And if the level of pricing will change, well, we'll cope with that. But if the value-based pricing in our industry is not recognized for the innovation we bring, then innovation will slow down. And I think that will be very negative for the whole industry, first of all for patients, because the very significant impact and advances which are made at this moment, which could lead to really transformational healthcare outcomes, will be staggered, and it will have an impact. This is not the first time in history that we deal with those discussions, but I think it only has led to a very positive thinking in industry that we even have to go for more transformational innovation. And I think that's going to be the answer. But if value-based pricing or innovation is not respected by a society as important, well, it will decrease and slow down innovation. Yeah. But we defend very vigorously the fact that the medicines we bring are value-based, and we discuss with authorities all over the world what the value is they want to pay for that.
Well, we started getting lots more folks standing up when I started talking about fecal transplants, so maybe you guys just want to change the subject. But let's start over here.
Yes, hi, I'm Fatima Núñez from Vall d'Hebron Hospital in Barcelona, Spain. I would like to tap into something that you've mentioned, and I think that's another paradigm shift that we're living with personalized medicine regarding how companies relate to each other. And this is regarding what you've mentioned several times during the panel, in that in order to offer personalized treatments, you have to put on the platter so many different molecules, probably cover a whole pathways that this involves. I think it should involve a very close collaboration, cross-company collaboration, that right now it's something that is being worked on. I don't think there is something standardized, or the companies have that sort of culture in terms of sharing with each other, you know, their molecules, their ideas, and their pipelines. And I'm saying this because now in Vall d'Hebron we are sort of leading a European project, basically, which is tasked with setting up the framework to do adaptive platform trials on 4 different diseases in Europe. And the one thing that we have realized is this is going to be a big hurdle. This will involve changing how companies talk to each other. And there are trust issues there, and there are legal issues there for them. And I think this is a shift that has to happen, and I would like to get your views on it.
And I'm happy to go start. So I entirely agree. I think it has to make sense. Some projects are better done in smaller groups, and some projects require bigger groups and even consortia to get them done, particularly if you want to sort of forge something together that is a true paradigm shift and that requires such an interdisciplinary come together. So when we started our first personalized cancer immunotherapy study, which was back in 2013, we actually saw that we couldn't do this on our own. So we created a European Union consortium that was also funded by the European Union. There were several companies joined together with different platforms and technologies, and 10 different academic centers, by the way, you may not be aware of that, including Vall d'Hebron, so your institution. And so we created the first personalized cancer vaccine trial in glioblastoma, which was recently published in Nature last year. But one lesson learned from that trial is that it's absolutely possible to create these frameworks when it makes sense and everybody understands what the role of each party is and what the contribution is to create these consortia. So I'm not sure we need novel platforms to achieve this. I think collaboration is fostered and driven just by needs, and when there are needs, companies collaborate and they share, and they can overcome all these legal and technical issues.
Well, I was significantly involved in the discussion of the platform trials because that was not just a discussion with industry, it was a discussion between industry and the regulators on how can we get faster to evaluating the new medicines. And so if you have a platform ongoing where you continuously can change the combinations which are tested in one and the same study, you could go much faster in evaluating different and it has been a difficult discussion, but at least we get going, and we try it out in a few disease areas now on can we get much faster get that done. And our team is instrumental in setting it up. There are other things we can do faster, is like what we do to Transliterate Biopharm, the pre-competitive collaborations where we can take out things in our industry to at least already work on the platforms we use to do clinical trials. The same quality system, same training system, the same protocols, the same way you do things, so that working together becomes much easier. And that basis is put in place. And then, of course, what is the content is collaborating on combination studies. One field where I think it is very advanced and very collaborative is in dementia, because if we don't work together, we are not going to make progress. Together working on biomarkers, and now there are 2 large cohorts ongoing, one in Europe, one in the US, which is set up already in order to follow patients for longer term, and different combinations can be studied in that. And the agreement is there between industries to test single agent, combination, so that we make faster progress. That's where there is a high need. If we don't do it there, in oncology is much more challenging to get companies but if it leads to faster approval and to better access very quickly, automatically people will start participating in it. So.
Go ahead.
One quick point. We also have in the background the scaling of these large clinicogenomic data sets that are coming together, which may also help derive future hypotheses for what combinations are going to be more effective than others. But I would agree, we have to, again, when we start testing more and more biomarkers and we find patients with multiple actionable ones and potentially even disease heterogeneity between metastases, we are going to have to treat with combination precision drugs.
Mm-hmm. Yeah, I was just going to, so I'm not affiliated with the company, so I can't talk about, address it in that way. But from a technical point, I might mention something that maybe is a compromise almost. So, you know, a chimeric antigen receptor T cell is a T cell that's been modified so that the business part of it is an antibody fragment to a specific target, whether it's an antibody to CD19 for leukemia or an antibody to BCMA for multiple myeloma or whatever it is. And so every time somebody comes up with a new CAR T, even for the same disease, with the same target, it's a different antibody. It can be to a different epitope. The efficacy can be different, the safety can be different, and then that's why we have this question of all these companies and all this money going into all these clinical trials. Well, there's a whole other sort of approach to CAR T, which is where the antibody portion is not against any particular target on anything that's in the human body. It's an antibody to, for example, a yeast protein that is not in the human body. So you make CAR T cells that are directed against a piece of a yeast protein. And so you put them into somebody and they do absolutely nothing. But then the person gets leukemia, so you give them an anti-CD19 antibody that has that piece of the yeast protein tag on it, which then engages with the surface of the CAR T cell, charges it with becoming specific to CD19. Okay, and then, so in other words, the CAR T cell is not specific to CD19, it's specific to a piece of a yeast protein. And you put the piece of the yeast protein on soluble antibodies that you just infuse into people. So then their leukemia goes away, and then they get brain cancer 5 years later. So they still have these CAR T cells, but you don't have to make new CAR T cells against brain cancer. It's the same yeast things, but you give them an antibody to EGFR variant 3 that has this tag on it. So those same CAR T cells now can attack that. So I guess my thought that just came to me when I'm sitting here is that you could have a company that makes these universal receptor CAR Ts. Then you can have all these other companies making the different antibodies that get infused, and then they don't work, they're gone in 3 weeks, and then you can just diffuse a different one. So you don't, so the big expense and all the testing is really, and the safety is with these, this universal CAR T cell, which is the same for any kind of, application.
That's really interesting. I have more questions for you on that, but I should get to the folks who are standing. Natasha, and then we'll go over here.
Okay, so I wanted to just pick up on this question about reference pricing, because when you import reference pricing, you're not just importing some random number that people have cooked up overseas. What you're importing is the sort of health technology assessments that other countries have come to determine value, and this is kind of something Scott Gottlieb touched on. What I find interesting is drugs like Kymriah and Sovaldi sailed into the NHS where we do quality assessments, and we have a really low rate of what we pay for our QALYs, our quality adjusted life years. We pay about 30,000 for it. So the question I have for all of you, and actually for you, Edward, is why, given that personalized medicine is inherently an argument about a better value treatment, why is reference pricing not an opportunity for your industry, for you? Why are you not sort of like praising values of health technology assessment and reference pricing? Because that could give you an advantage against all these other treatments. That's my question. And to the panel as well, obviously. Is it not an advantage?
That's what they call a hospital pass in rugby.
Yeah. Well, personalized medicine should deliver more value because it's much better outcome driven based on specifically targeting, and therefore it could be much more valuable to the healthcare system, but the cost to do it is also significantly higher because typically you need to diagnose, or you need to determine 100 patients and probably 20 will get the therapy, and therefore the additional cost of getting to personalized medicine, not many societies are prepared to pay for that. So it is not obviously that it becomes cheaper. The outcome could be better, but there is a lot of pluses and minuses on how to implement personalized medicines on a large scale. In cancer, if everyone now is going to be sequenced, the tumor is going to be sequenced and the mutations will be known, and it will be very known how to combine different therapies, then it becomes routine. Before that's happening, every drug, like we introduce now, Balversa for bladder cancer, we'll have to develop. We are developing our own assay, and every patient which has now bladder cancer is diagnosed before, yes or no, he or she will respond to the therapy. So the value proposition of personalized medicine is still to be worked out. If you go to the NHS, you have to have the discussions. The value proposition has to be worked out there. Better answer than me? No? Please, Anne.
Thanks.
Take it on.
So I've been working in Africa for 22 years, and I have a background in infectious disease, but I'm not going to ask a question about stool. So I think it's really interesting that when you look at, or you talk to drug companies, which I've done a lot of, with regard to, for example, access to drugs for patients in Africa around cancer, the pricing is not the issue, even though all of the companies in the US and around the world that produce immuno-oncology drugs try to agree on a price within 3 to 5% so that insurance companies will pay them for that. When you talk about delivering that drug to Africa, the idea of GDP-scaled pricing or reducing pricing for those patients is not the issue. The issue is actually the problem with adverse reactions, because the infrastructure of those healthcare systems won't support those patients if they have a challenge, same for CAR T, same for anything else. So when thinking about pipeline, when you're developing drugs or thinking about new personalized medicine therapies, and you recognize that that drug will require infrastructure beyond, if you think about HIV paradigm, you can give anybody HIV drugs anywhere in the world. Thinking about that paradigm, what would the role be for pharmaceutical companies in developing these drugs in the infrastructure of new markets? In other words, the market in Africa or South America or Southeast Asia isn't as developed because that infrastructure isn't there for your drug to be there. But is there a need for investments in that infrastructure so that those markets are then created and therefore the whole world can benefit from these personalized medicine therapies?
Great question.
Well, is it their role for us to invest in the infrastructure? I think the best solution to start with is evolve as fast as possible to simpler solutions which need less infrastructure. And because that's what we learned with HIV. In the beginning, multiple medications, very complex. Now, one pill once a day, 2 or 3 pills are distributed on a very large scale, and nurses and even very simple infrastructure can administer it. The cost of getting to oncology therapy in places where I have been active, in Congo, in Rwanda, in Central Africa, is let's say, dealing with infectious diseases already first, dealing with the cesarean sections is next. Can we get to clean surgery and making sure that the surgical outcomes are good? So it will take a lot of time before the infrastructure can be there to go to CAR-Ts. Yeah? But it won't take a lot of time to get to the simpler small molecule oncology drugs, which could be available. We have a product, Zytiga, for prostate cancer, which is a simple pill, an oral pill. You could think about going faster on developing that. But is it up to us to globally build the infrastructure? I think we should partner with the local with the countries as well as with WHO to start working on how can you see first types of oncology drugs being implemented. Small molecules, maybe chemotherapy, very challenging, as you say, dealing with side effects. Maybe the next step, very effective antibody therapy could be the next stage. But it is a very high hurdle. And if you deal with a country and where many have $10 to $20 per year per person for healthcare, you have, as a country, as a public health person, you have to introduce what's most important for that country first. And typically it is, let's vaccinate, let's have the most basic things in place, because there is no more resources than that. Hygiene, clean surgery, making sure that sterile surgery can be done so that you don't cause more problems. And it's much more, well, it's a very complicated, long-term, but we need to be very determined. If we one day want to get to global cancer therapy, we'll have to do that. But the first solution is, in my opinion, the simplest possible therapy. And can we get to not the first generation therapy, but the fifth or the sixth, and then make it cheaper, much more available, and simple to implement. But it's a comprehensive approach to public health in the world.
I've got more questions for you guys on that, but I think let's go back to the audience, then we'll return to the topic, maybe.
Thanks. I'm Bo Bai from Foundation Medicine. So as the panelists mentioned, the most drug in development is with biomarkers. But there are cases that the clinical trial design is a user primary endpoint as based on biomarker to ensure success, then use second endpoint to get the all-comers label, or use second trial to get all-comers label. And is that makes sense from a business perspective. In that case, if a drug get all-comer label for one specific indication, it seems the company is less incentivized to identify a subset of patients that may benefit more based on biomarker identification. So in that case, do you think that kind of impedes the development of personalized medicine? And what is the company's approach?
I could take a quick stab at just that from a more macro level. I mean, I think you're right. There are drugs that have labels that are with and without biomarkers in different indications. And I think that, simpler is probably better. But I think as we see, if more and more drugs are launching with biomarkers, they're going to have higher and higher efficacy hurdles that the next drug is going to have to meet. And so enrichment strategies start to make sense. So I think it's going to be a moving target, primarily. But I understand your point. I mean, I think it would be simpler going with all comers. I think we're just going to start to see fewer and fewer of those. I would say just also what's complex for clinicians, if we just talk about the checkpoint drugs, they can't keep up with the different labels by tumor type, by line, by drug, by biomarker. They don't. It's becoming impossible for them to keep pace with it. So I think there will need to be standardization. And then, by the way, biomarkers don't have to be just for therapy guidance, right? We're thinking about solid tumor MRD and tracking response. And so there will be other biomarker strategies that are going to be relevant, even if it's not specifically for a therapy guidance decision.
In my view, I mean, definitely biomarkers will continue to play a role, particularly for medicines that are broadly applicable, like checkpoint inhibitors, immunotherapy. we also see a paradigm shift, particularly with more innovative treatments. So in our case, for instance, we do not start all comers and then try to find a biomarker. We actually develop a drug based on the biomarker. So we first look at the target, the cancer target, look at the natural range of the copy numbers that this target occurs, and then create a T-cell receptor that ideally fits to that biomarker, and then only apply the T-cell receptor actually in patients that have a sufficient copy number right up front. And I think that's really turning this whole system around, starting with the biomarker, having the understanding of the biomarker set as the first point in your development, and then developing the drug actually based on that biomarker. And I think we'll see both approaches, but the second one is something that we'll see more about in the future.
What's J&J's philosophy on it?
Well, my philosophy is that you have to try to determine the best possible outcome for your drug using the biomarkers and even selecting the patients, because if you don't do it, somebody else will do it. And so you better do it yourself in clinical trials to make sure that you know for which drug for which patient the drug works best. Because very soon afterwards, you will have a competitive medicine coming in. And so if you have not the highest possible efficacy for the specific population, you might lose the drug very quickly. Going broad with no, and it only works in half of the patients, it will not turn out very well in the long term. So it's much better to do the scientific, rigid, and good strategy in order to determine who responds and who doesn't respond, and market it for that.
That point, sorry, just really quickly, your point is absolutely, when we see it in the data, first comer to the indication doesn't need a biomarker, and they're competitive right behind it is going to pursue a biomarker strategy and to enrich their signal. I think
It's
we're
kind
seeing
of what happened with Merck and Bristol Myers, right? It
and
initially appeared Bristol had taken the better approach by not having such specific biomarkers, and then it turned out the opposite, right?
Yeah, and it's going to keep going.
No
Absolutely.
way to hide.
Yeah.
Thanks for the panel today. It's a great discussion. I have two quick questions, if I may. The first one is related on consent a little bit, and then the other one's on pricing. So firstly on consent and CAR-T, many of us know the story of Henrietta Lacks and HeLa cells. I am presuming that universal CAR-T cells will come from cancer patients, potentially deceased cancer patients. How do you think their families will feel about that if you're not fully informing them what you're doing when you're storing these cells at processing facilities?
Is that for me?
Sure.
The cells that we collect from a patient are only used to make a drug for that patient. If you're talking about universal, are you talking about universal, like allogeneic CAR T cells? Mm-hmm. Right. So those kinds of the way that probably will be structured, because it doesn't exist right now, is the way is from donors who voluntarily donate, knowing what's going to happen with their cells. It's not from patients who unwittingly had some extra material left in the freezer. Perhaps that's the HeLa story, that were then used to generate new drugs or used to treat patients that you charged for the treatment. So I mean, we would never. So the plan would never be to make CAR T cells from patients and use those to treat somebody else. The allogeneic thing is from a healthy donor, just like people donate at the Red Cross. The entire blood system, it's a voluntary blood system in the United States. No one gets paid to donate blood, but blood's used and patients get billed for the transfusions and so forth. Does that answer your question?
Well, doctors, go ahead.
Yeah. Kind of, I think it's much more complicated than that, and it requires a bit more thought. Blood is very different from cells and CAR T cells. Blood doesn't have genomic DNA. It's not immortal. It's just one use. If you're creating CAR T cells, universal CAR T cells, that then becomes a billion-dollar drug, and the patient thinks that they know what they're doing when they're donating cells.
But again, patients are not donating cells for anyone other than themselves.
Well, so Dr. Singh, how is it working with your program with the off-the-shelf approach that you have, with directed T cells that you have already from, I assume, from a patient that had that kind of cancer?
Just confirming what Dr. Seal said. So in our case, and this is still preclinical stage, we have not applied this to cancer patients. The source for the allogenic material are healthy donors. They come to the hospital just as they donate blood, and they have an informed consent form that actually fully informs them what's going to be done, and they donate those cells at this moment just for research purposes. At a later stage, then they would donate that for the manufacturing of multiple, ideally thousands of doses that are then stored and frozen and ready for application. But it's always healthy donors, and there are scientific reasons why we use this. This is not a legal aspect. There's scientific reasons why we use healthy donors, because they typically have a better state of their immune cells that we can tap into.
My understanding is also there's a need for these donors to be well characterized and recallable. If you're process developing around one donor and then you run out of those cells, and then you have to find a new donor to do it again, I think it's going to be, there's a lot of questions to be answered around allogeneic. But in this donor base and managing the donor base, and then I think the point, if you start editing, is it a different editing approach depending on who the donor is? Which means, does that then again create scalability? You were mentioning, Paul, earlier about the CRISPR editing of donor cells. My question is, I guess, if you're not going to have one donor to treat everybody, so does it have to be, do you have to then start editing differently for each of the donors one at a time, or is there a universal editing approach?
No, I think, I mean, there's a million kinds of things that could happen in the future. The thing I'm speaking about is, would be the same editing approach. For what we're doing now, it would be the same editing approach with the same reagents, the same process, regardless of who the donor is. In other words, people have PD-1 on their cells. And if you're developing a therapy where you don't want the cancer to be able to shut off the cells, then we would use CRISPR to delete PD-1 from the cells. And the way you would do that would be the same no matter where the cells came from.
Your next question?
Thank you. So there's around about 50 very expensive gene therapies that are late stage or approved between Europe and the US right now. Average price is $500,000 to $1 million each per person per treatment. Healthcare budgets are maxed out for a lot of jurisdictions, especially our jurisdiction also. Do venture capitalists realize, and pharmaceutical companies realize that this is not sustainable? Are they actually engaging in a socialist experiment? Are they venture capitalists becoming socialists, and they don't expect to get paid for these clinical trials and these therapies? Or is there some better business model that you're actively thinking about?
Well, no, we won't be there anymore next year if we don't charge for our medicines. But there should be, and there could be, different models which again is outcome-based and value-based, is how long therapies would work, and could there be a payment system over years for the real benefit over years if it is a one-time gene therapy. I'm not an expert in payment systems, but we think in our organization that we should not stop advancing the science. We should start building new business models. And the other aspect is more global and faster access to the new therapies, not just in a very small part of the world. China is now starting to reimburse medicines on a large scale for innovative medicines. So the Western world, between the countries who can pay and who have the ability to pay, and new business models, globalization and new business models should allow for much better access at more reasonable pricing. But it's a pathway we have to go through in the next few years.
Do you agree with the premise of the question that you will end up not getting these new one-time therapies paid for at the current pricing and business models?
Not be paid for. I think we are not into that, so I'm not an expert in the area. But I think as long as the benefit is significant, the current society will find a solution to treat orphan diseases in one or another way. It will be very challenging. It will be giving in from companies, giving in from society, but at the same time also new business models. And think will depend on the innovation it brings.
I think it does highlight the need to just be able to characterize the economic impact of an innovative drug across or an innovative portfolio of drugs across the entire patient journey, and not just one episode of care. And it's just a simple point, and I don't think we've necessarily done a great job of being able to do that, because often the healthcare information is siloed and it's not interoperable, and so we can't actually really tell what the actual total journey looked like for a patient who went down a precision arm versus one who used a more standard arm. But I think as those data sets come out, it will actually show that we are actually providing cost-effective treatment, but you're taking costs from one bucket and moving it elsewhere.
So I can't speak from a company point of view of sustainability, but as a hospital where our physicians prescribe the commercial CAR Ts, it's I think in general a money loss at this point for the hospital. And I tried to speak to someone before coming here to try to understand the whole billing process, and it's impossible to understand. Partly because it keeps changing, but one of the things that is pretty clear is that as these drugs, we get more experience with them and we understand safety and so forth, things will happen to make it less expensive. So just to give a quick example, the Novartis drug is a complete outpatient process. The collection of the cells is an outpatient. The patient disappears. The cells are manufactured. The patient comes back actually to my apheresis unit, an outpatient facility where the cells were originally collected, and they sit, and for 10 minutes, about 2 ounces of CAR T go into them, and they're observed for an hour, and then they go home. And if they develop CRS, cytokine release syndrome, then they may come back and get admitted. But it's mostly an outpatient procedure. That is the economics of that for the hospital is completely different than the Kite Gilead product, which is an inpatient infusion and inpatient stay. And so when things are done on outpatient side, hospitals get reimbursed better than on inpatient side. And so that, what I'm saying is as we learn more about these drugs, I think costs will come down for the hospitals that use them.
That was a fascinating question. Go ahead.
Hi. Thanks to the speakers for being here. It's been really enlightening so far. My name is Amy Wessel. I'm from Institut Pasteur in Paris. And I had a question about, basically, we hear a lot about omics, so using DNA, RNA, proteomics, all of these personalized approaches that are on that line. And I haven't heard as much about live cell-based assays. So actually taking an ex vivo from a patient and then testing the cells directly on a drug, for example. Not saying that proteomics or DNA and RNA are not valuable, but I want to hear your side of what you all do with cell-based assays, because it's clearly important for IO drugs and for immunotherapies to be testing cell-cell interactions. And if there are bottlenecks for you, or if you find it not as interesting as proteomics or transcriptomics, for example, I'd love to hear that commentary.
I mean, we have started thinking about that right now, less from an efficacy point of perspective, but more from a safety perspective. So we're thinking about creating warehouses or libraries of many different T cell receptors that would then be transduced into autologous T cells. And those T cell receptors potentially could have off-target toxicities, meaning that they cross-react with something that is just occurring in that particular patient. And so this is really hard, given the diversity of the HLA system, to really predetermine. So one potential way out of that would be to actually develop a simple blood-based assay where you incubate your TCR-T cells that could be cross-reactive with the blood cells of the patient as a release assay. So that's probably a relatively low-cost but straightforward way to determine, or at least decrease, some of the safety risk. We have also done a clinical trial in Europe in glioblastoma patient, where before applying peptides from a library of 80 different predefined peptides for cancer vaccination, we've first actually checked in the individual patient which of these peptides are actually immunogenic in that particular patient, and only vaccinated those agents that were immunogenic in that patient. So we've explored this. I think this is an absolutely viable approach that doesn't replace all the basic science and the omics, what you refer to, that we have to do to determine what the right drug candidates are, but that allows for certain specific circumstances to optimize the regimen.
There are certain labs, clear labs in who do that type of testing in oncology, which will collect cells, grow them, and test the combinations of different cancer. But it's a very complex process. It's lab-developed test. It's very difficult to, let's say, industrialize and bring it all, because it's not something which is like standardizable, get approved by the FDA and send around. But it's I can't tell, I'm not an oncologist, but there is definitely that testing ongoing. Whether it will be ever generalizable is something else.
Do we have time for one last question? Yeah. Okay.
Hey, thanks. Mark Watson from the Guardian Research Network. We have two passions. One is to bring clinical trials to patients in the community that don't always get access to those, but a big passion of ours is ultra-deep clinical data and innovative clinical trial designs. And I wondered what the panel thought about the concept of synthetic controls and sort of designs to do trials faster. I guess a little preface, Tufts down the road here did a study on drug development cost, and to my estimation, about half of the $3 billion or so was preclinical. In my humble opinion, that's probably not going to change because that's just hard and heavy chemistry and work, but the other half came from the clinical side. You know, what if we had a paradigm shift in clinical trials where you could test a lot of personalized drugs fast with a synthetic control population and only give drug to 20, 25 patients and get a signal? I'm just curious what the panel thinks about trial design innovations.
So I think maybe someone here can give a more directed answer to your question, but the way I see something like that being helped is a program that we've started, which is companion animal CAR-Ts, treating dogs. So it's a paradigm shift. It's cars chasing dogs. Yeah, so the idea is that dogs develop cancers that are very much like human cancer, much more so than mice that you put stuff into. Dogs have intact immune systems. They've had the same types of treatment that humans have had, radiation therapy, chemo. They undergo apheresis to collect their T cells, and we're developing ways of making CAR T cells. And I think that there's a million different possibilities of all these different kinds of CAR T constructs, targets, different antibodies to the same targets. And it turns out that there's a lot of homology between canine targets and human targets. And the same exact construct could be used for treating a dog that you would then directly put into a human. And then I think, and these aren't research dogs that we're using so there's a vet school with Penn, and we're working with that. These aren't dogs from shelters. These are dog patients whose owners sign informed consent. And so I think a lot could be learned by treating dogs that need treatment, and that can be directly applied to humans, and it would be a lot less expensive.
I would like to answer that question. I think you bring up probably some of the most important transformations we have to do. Industry started to share placebo data. For example, everyone who has done a psoriasis trial, the next one, could you use the controls from another company? And so the placebo data sharing is already ongoing. The FDA is very much encouraging that. Synthetic controls, like you say, is ongoing. It's very good for patients because then only 1 in 4, 1 in 5, 1 in 3 instead of 1 on 1 will receive control, placebo or control. That's where trials become much more attractive, will go much faster, and will cost lower costs. The other area is, will it be possible, question mark, to do clinical trials in the real world through electronic medical records? Can you randomize patients in the clinical healthcare system, because now data are collected so comprehensively that you don't have to do dual work? Yeah. So what we do in the industry for the last 50 years is we copy the data from the record, put it in another record, and then we submit it to the FDA. So if you can use the healthcare record as the source for the information, and you just plug in there and you can tap the data, the cost will be hugely reduced, because you don't have to do the monitoring, the quality control, etc., on the transcription of the data. So the field of doing clinical trials is ripe for big disruption with what you are saying on synthetic controls, but also many other things. We are now going to do the first complete virtual trial with studying atrial fibrillation with the Apple Watch and telephone, and people won't even see the physician. Yeah? So we are going to start learning on how can we do trials completely virtual in the population. But it's a big paradigm shift, but it is such an important one, because it will decrease cost of development and increase speed to result, and that is a big paradigm shift.
It's extremely critical for rare things, particularly rare cancers, rare biomarkers, where just the time to find that exact patient to then give them placebo doesn't make sense. And so I would very much agree that these, also when we see these real-world data sets that are scaling, you have really good reference points. But as we see the fragmentation of oncology by biomarker, by this, by that, we may actually have to, we may require synthetics to actually enable the development across the whole field, because we're just not going to be able to enroll what we need to enroll otherwise.
I think we've reached our time. I want to thank everybody for your great questions. You made my job very easy, and thank you to our panel.
Thank you.
Thank you.
We spoke on the phone last week.
So we're going to adjourn for lunch now and come back for the Harvard Business School case study. And Professor Habermatch is saying that if you haven't read the case, you should read it at lunch. But you can come back anyway, even if you haven't read it.